In the rainforests of Thailand, on the underside of a leaf approximately 25 centimeters above the forest floor, there is an ant that has been dead for 3 days. It does not know this yet. It is still walking, not wandering, not stumbling, not moving in the erratic way that dying animals move when their systems are failing and their coordination is breaking down. Walking with purpose, along a path it did not choose, toward a location it has never visited, driven by something that is not hunger and not instinct, and not any impulse that originated inside its own nervous system. Its mandibles are locked around the central vein of the leaf, beneath it, in a grip so precise and so powerful that researchers who first encountered these ants in the field assumed the jaws had been fixed there by some external adhesive. They had not. The ant locked them there itself. In the final minutes, before whatever remained of its neurology ceased to function, it bit down on that exact location, at that exact height above the forest floor, in that exact compass orientation relative to the surrounding vegetation structure, and it held. Then it died. And then something began to grow from its head. The thing growing from the ant's head is a fungus, not a contamination that settled opportunistically on a dead body the way mold settles on rotting food, not a secondary infection that moved in after the ant died of something else. A fungus that spent the previous 2 to 3 weeks living inside the ant's living body, moving through it deliberately, consuming select portions of it with a precision that left the parts it needed intact, and metabolized only what it did not, threading its cellular network through the gaps between the ant's own cells and around its muscle fibers, and the entire time issuing chemical instructions to a nervous system it had commandeered so completely that by the moment the ant bit down on that leaf vein, not a single behavioral decision the ant appeared to be making had actually originated within the ant. Everything the ant did in those final weeks, every step it took in a specific direction, every surface it descended toward rather than away from, every turn it made in the forest understory, every moment of movement that brought it closer to the precise microhabitat the fungus required. All of it was not the ant deciding. All of it was the fungus directing. And the fungus has no brain. It has no nervous system, no eyes, no ears, no sensory organs of any kind that biology recognizes as capable of perceiving the forest environment through which it was navigating the ant's body. It is, in the most fundamental biological sense, a network of cells, nothing more, and yet it directed an animal with a fully functional nervous system, refined by 100 million years of ant evolution, through a sequence of behaviors more spatially precise and more temporally coordinated than most animals ever perform when acting entirely on their own initiative. Understanding how this is possible is one of the most genuinely unresolved questions in all of modern biology, and the story of how researchers have spent decades trying to answer it takes you into places that the word biology does not fully prepare you for. The floor of a tropical rainforest is one of the most competitive environments on Earth. The soil alone in a single square meter of Thai rainforest contains more individual organisms than there are human beings alive today. Every fragment of organic matter, every source of nutrition, every unit of space that can be occupied is contested simultaneously by a diversity of organisms so vast that ecologists studying it spend entire careers mapping only small portions of it. In that competition, ants have become one of the dominant forces. Not because individual ants are physically powerful. A single ant is one of the least threatening organisms in the forest, but because ant colonies operate as unified superorganisms with collective capacities that no individual ant possesses. They have become among the most successful animals in the history of life on Earth. Camponotus carpenter ants, the species most commonly targeted by Ophiocordyceps unilateralis in the forests of Thailand and surrounding regions, build colonies that can house several 100,000 individuals. The colony coordinates, foraging across territory that covers hundreds of square meters, manages disease within its population, through sophisticated, hygienic behaviors that include identifying sick nestmates, quarantining them, and removing them before they can spread pathogens to healthy individuals. It conducts warfare against competing colonies with strategies that involve advanced scouting, coordinated assault, and tactical retreat. It has been doing all of this for approximately 100 million years and has solved problems during that time that human engineers working with modern computational tools have found instructive to study. The zombie fungus encounters this superorganism and is entirely indifferent to everything about it except one thing. It moves through the forest at the height and density the fungus needs to complete its reproductive cycle. To Ophiocordyceps, the ant colony, with its 100 million years of collective intelligence and its hundreds of thousands of sophisticated individuals, is a delivery mechanism. Each ant is a vehicle, a warm body moving through the forest at the correct scale, in the correct habitat, at the correct times of day, to serve as transport for a fungus that cannot move on its own and cannot reproduce without reaching a very specific location with very specific environmental conditions. Everything the ant does, everything the colony has evolved to do over 100 million years, is from the fungus's perspective, simply the operating behavior of an available transport system. And the fungus has learned, over approximately 48 million years of its own evolutionary history, how to take control of that transport system with a precision that has left every researcher who has studied it reaching for language that biology does not normally use. The infection begins at the surface of the ant's body, in a moment so small and so quiet that nothing in the surrounding forest notices it happening. Ophiocordyceps spores are released from the elongated reproductive stalks that grow from the heads of previously infected dead ants. These stalks, the ascocarps, produce spores continuously during their active period and release them in dispersal events, timed to coincide with periods of high ant traffic through the understory vegetation below. The spores are not heavy, they drift. They fall through the layers of the forest in a dispersal that is invisible to any eye and undetectable to any sense the ants possess. They land on the bodies of foraging ants moving through the vegetation beneath the infection sites. They adhere to the surface of the cuticle, the hardened outer layer that protects the ant's body from the environment, and then they begin dissolving it. Ophiocordyceps spores produce a suite of enzymes immediately after attachment that break down the chemical bonds in the cuticle, creating a pathway through the ant's outer armor. This penetration process is slow by the standards of what follows. It takes hours. During those hours, the ant continues doing exactly what it was doing when the spore landed: foraging, communicating with nestmates, following the pheromone trails laid by other workers, performing whatever tasks the colony's current collective priorities have assigned to it. The ant has no mechanism for detecting that its own outer surface is being chemically dissolved by something approximately 1/100th of a millimeter in diameter. Once through the cuticle, the fungal cells enter the hemicole, the body cavity that in insects serves a function analogous to the circulatory system in vertebrates. The hemolymph, the clear fluid that fills this cavity and bathes the ant's internal organs, carries nutrients, immune cells, and chemical signals throughout the body. The fungus enters this system as individual hyphal bodies, single fungal cells that look under a microscope, like small rounded packages drifting through the hemolymph currents. And then it makes its first set of choices. This is the moment that researchers studying the early phase of Ophiocordyceps infection find most difficult to account for within conventional parasitology. A fungus entering a host body has, in almost every other documented parasitic system, one primary drive: consume available resources as rapidly as possible and reproduce before the host's immune system eliminates the infection. Ophiocordyceps does not do this. Instead of beginning indiscriminate consumption of available tissues, the hyphal bodies distribute through the hemolymph and begin selectively targeting specific tissue types while leaving others entirely alone. The fat bodies, the ant's energy storage organs, are consumed early. These are rich in lipids and proteins and are not immediately essential for locomotion. The hemolymph itself provides nutrition. Various non-essential soft tissues are metabolized progressively, but the muscles that control the ant's legs and mandibles are left functional. The nervous system is left structurally intact. The sensory organs that allow the ant to perceive its environment continue operating. The fungus is not eating its host, it is managing it, maintaining the functionality of the systems it needs while consuming the systems it does not. It is performing at the cellular level, the kind of triage that a mechanic performs on a vehicle that needs to keep running while being serviced. Keep the engine working. Everything else is available for fuel. For the next 2 weeks, the colony around the infected ant observes nothing unusual. This is perhaps the most remarkable aspect of the early infection phase. Not what the fungus is doing inside the ant, but what it is preventing from happening outside it. Ant colonies are extraordinarily sensitive disease detection systems. The chemical signals that colony members exchange constantly through touch, through grooming, through the shared chemical environment of the nest, carry information about individual health status that the colony's collective immune response uses to identify and quarantine sick individuals before infection can spread. The hygienic behaviors of Camponotus colonies are sophisticated enough to detect and respond to a range of bacterial and fungal pathogens at concentrations that no external observer could identify without laboratory analysis. Ophiocordyceps evades this system for 2 full weeks. The mechanism that allows it to do so is not fully characterized in the published literature, which is itself a remarkable statement given how intensively the system has been studied. The leading hypothesis is that the fungus produces compounds that mimic the cuticular hydrocarbon profiles that ants use to identify nestmates as belonging to their colony and as being in normal health, by chemically impersonating a healthy ant from the surface of an infected one. The fungus may be speaking the colony's own recognition language, producing the chemical signal that says all is normal here in a system that has evolved to listen for exactly that signal. If this hypothesis is correct, it represents an evolutionary achievement of extraordinary sophistication. The fungus has not just learned to infect carpenter ants, it has learned to speak carpenter ant, to produce the molecular vocabulary of ant colony health assessment, and use it to buy the 2 weeks of undetected internal growth it needs before beginning the behavioral manipulation phase that will bring everything together. Then, in the 2nd or 3rd week of infection, the ant stops being itself. The first sign is timing. The infected ant begins leaving the colony outside the coordinated activity periods that the colony's pheromone signals organize. Healthy forager ants in Camponotus colonies operate within rhythms set by the collective chemical communication of the colony. They are most active during specific temperature windows and respond to foraging signals laid by scouts. The infected ant begins moving outside these rhythms, leaving the nest during the midday heat peak when foraging ants normally shelter, returning at irregular intervals, disrupting the predictable temporal pattern of its own behavior in ways that researchers observing colonies in infected forest areas have learned to recognize as one of the earliest visible signs of late-stage Ophiocordyceps infection. The second sign is movement quality. The infected ant's locomotion becomes convulsive. Muscle spasms interrupt the smooth, coordinated 6-leg movement that characterizes healthy ant movement. The ant loses its grip on surfaces it would ordinarily cross without hesitation and falls. It rights itself, falls again. The mechanical smoothness of normal ant locomotion, the product of a neural muscular coordination system refined over 100 million years of evolution, breaks down in ways that look like a hardware failure rather than a behavioral choice, because that is exactly what they are. The third sign is direction. The infected ant moves down. Healthy Camponotus carpenter ants in the Thai rainforest spend the great majority of their active time in the upper layers of the forest vegetation. Colonies are typically located in elevated dead wood several meters above the ground. Foraging occurs primarily in the canopy and middle story of the forest structure, where food resources are concentrated. Movement into the forest understory, the zone close to the ground, where humidity is high, light is low, and temperature is more stable, is not part of the normal behavioral repertoire of a healthy forager returning to the colony. Infected ants go there anyway, and they do not go there randomly. The descent is directional, consistent, reproducible across infected ants from different colonies in different sections of forest, across different field sites in Thailand and Brazil and the eastern United States. The target is always the same narrow band of the forest understory, 20 to 25 centimeters above the forest floor, where the combination of temperature and humidity creates conditions specifically optimal for Ophiocordyceps spore germination and successful dispersal onto passing ant traffic below. How a network of fungal cells directing an ant's muscles achieves that level of navigational precision was the central question that David Hughes at Penn State University and his colleagues set out to answer in research that culminated in a landmark study published in PLOS One in 2011. Hughes's team used a three-dimensional imaging technology, specifically microcomputed tomography scanning, to examine the internal structure of infected ants at multiple stages of the behavioral manipulation process, looking for the anatomical mechanism that produced the behavioral precision they were observing in the field. What they found inside the ant's head was not what they expected. The brain was largely intact. In specimen after specimen, the neural tissue of the ant's brain showed minimal fungal invasion. The neurons were present. The neural architecture was structurally preserved. The part of the ant that the ant uses to process sensory information and coordinate behavioral responses was not where the fungus had concentrated its activity during the manipulation phase. The fungus was in the muscles. A network of fungal filaments had grown throughout the ant's body and positioned itself specifically around the muscle fibers responsible for locomotion. Not replacing those muscle fibers, not consuming them, surrounding them. Each individual muscle fiber was encased in a network of fungal cells, maintaining intimate physical contact across the entire muscle surface. And from that position, from the surface of the muscle fiber itself, the fungal network was releasing compounds that caused the muscles to contract and release in sequences that bypassed the nervous system entirely. The ant's legs were receiving movement commands from the fungal tissue wrapped around its leg muscles. Its mandibles were responding to signals produced by the fungal network surrounding its mandibular muscles. The nervous system was structurally present and biochemically functional, but it was no longer in charge. The motor commands were coming from outside the neural architecture entirely, from a parallel control system that had inserted itself at the level of the muscle fiber and rendered the brain's own instructions irrelevant. Hughes described this finding as requiring a fundamental reconsideration of how behavioral manipulation in parasitic systems could work. Every previously documented case of parasite-induced behavioral change operated through the host nervous system in some way, toxins targeting specific neurotransmitter systems, neural tissue invasion, chemical analogs of host neurotransmitters flooding the circulation. Ophiocordyceps had bypassed the nervous system entirely. It had gone around the brain and spoken directly to the muscles. It had found a way to control movement without touching the controller. The brain was still in the ant. The decisions had been outsourced. But the muscle control mechanism, as extraordinary as it is, explains how the ant moves. It does not explain where. Producing muscular contractions that move an ant's legs is one problem. Producing muscular contractions that consistently navigate an ant from wherever it happens to be in the forest to a leaf surface at 25 centimeters above the ground, in the specific microhabitat with the temperature and humidity profile Ophiocordyceps requires, is a fundamentally different and vastly more complex problem. Researchers studying Ophiocordyceps across multiple forest sites have measured the height of dead infected ants repeatedly, and with instruments precise enough to distinguish differences of a few centimeters. The results cluster at the target height with a statistical precision that eliminates chance as an explanation. The ants, from different colonies, infected at different times, moving through sections of forest with different vegetation densities and different topographic profiles, all die at the same height. The navigational accuracy of the fungal system is real, and it is consistent across the entire geographic range of Ophiocordyceps infection. The hypothesis that best accounts for this navigational precision proposes that the ant's own sensory systems are still operating during the manipulation phase and are providing the fungal network with the environmental information it uses to direct movement. The ant's temperature sensors, its humidity receptors, its light sensing systems, its proprioceptive awareness of its own position and orientation in space, continue functioning because the brain that integrates them is structurally intact. The fungus produces compounds that alter how the ant responds to the signals those sensors generate, making specific environmental gradients feel like signals to approach rather than signals to avoid or ignore. The ant's perceptual system becomes a navigation instrument pointed in the wrong direction. Its 100 million years of evolved sensory sophistication, refined to serve the ant's own survival and the colony's collective needs, is turned toward a destination the ant would never have chosen for itself. When the destination is reached, when the correct leaf surface at the correct height, in the correct microhabitat has been found, the fungal network issues its final instruction. The mandibular muscles receive a signal that produces a contraction the ant cannot release, not a voluntary bite, a tetanic lock. The maximum force the mandibular muscles can produce, held continuously, without the relief of release that voluntary muscle contractions always eventually allow. The bite force applied at this final moment has been measured as exceeding the maximum voluntary bite force healthy ants of the same species can produce. The fungus, in its final act of motor control, commands a grip stronger than anything the ant can achieve on its own. The mandibles close on the leaf vein. The ant dies. The grip holds. And over the following days, the reproductive structure of the fungus grows from the back of the ant's head. The ascocarp extends upward through the space where the ant's head capsule was, elongating toward the air currents that move through the forest understory, timing the release of the next generation of spores to coincide with the peak foraging activity of the ants moving through the vegetation below. Above the zombie ant graveyard, in the canopy, where the colony's normal activity continues, something has been happening that the colony has not been passive about. The ants can identify infected nestmates. Not in the early phase, when the infection is invisible by every measure available to the colony's detection systems, but in the late manipulation phase, when the behavioral anomalies have begun and the infected ant is moving differently. Healthy nestmates respond to it with a specific behavior that has no equivalent in their treatment of healthy colony members. They carry it away, they pick up the infected ant, transport it from the colony's active territory, and deposit it at a distance in the forest vegetation, not killing it, not grooming it aggressively the way they groom nestmates, contaminated with non-lethal pathogens, removing it with a calm deliberateness that researchers observing the behavior describe as almost mechanical in its consistency. The behavioral response is calibrated to get the infected individual away from the colony before it dies in a location where spore fall could reach the colony's foraging routes. What the ants are detecting in the infected nestmate during this removal response, what specific chemical signal at what concentration triggers the carrying behavior, rather than the grooming or killing response, is one of the genuinely open questions in the Ophiocordyceps literature. The detection is happening. The timing and precision of the detection, identifying infection at exactly the stage when removal is most protective of the colony, is not achieved by any instrument researchers have built to look for the same signal. The ants are more sensitive to something about the late-stage infected nestmate than any technology directed at the same question has yet managed to be. The oldest confirmed evidence of this interaction is a fossil, found in amber from the Messel pit in Germany in 2010 by David Hughes and colleagues, dated to approximately 48 million years ago. The amber does not contain the fungus, it contains a leaf, and on that leaf, a bite mark, positioned on the central vein, at a location and in an orientation indistinguishable from the bite marks left by zombie ants dying in the Thai rainforest today. 48 million years ago, in a European forest that has long since become something else entirely, an ant bit down on a leaf at the direction of a fungus that had spent several weeks living inside it. The ant died. The grip held. The spores fell onto the ants, moving through the understory below. The next cycle began. That cycle has not stopped. It has been running continuously through 48 million years of climate change and continental movement, and the appearance and disappearance of countless animal lineages around it. It was running when the ancestors of modern horses were small forest animals the size of dogs. It was running when whales still had legs. It was running when the ice sheets of the last glacial maximum covered the northern hemisphere, and when they retreated. It incorporated every generation of ant colony sophistication as new selection pressure that drove the fungus to greater precision. Every advance in the fungal manipulation system drove the colony's detection response to greater sensitivity. 48 million years of arms race, producing in both organisms a level of sophistication that continues to exceed what the researchers studying it can fully explain. Hughes, who has devoted more of his research career to Ophiocordyceps than any other scientist, said in a public lecture in 2019 that studying the zombie ant fungus had fundamentally changed how he thought about the boundaries of what biological systems were capable of, not because it was the most extreme example of any single capability, but because it combined navigational precision, behavioral control through a novel mechanism, timing sophistication, host-immune evasion, and colony-level evolutionary arms race dynamics into a single system that had been running and refining itself for longer than most animal families had existed. In the Thai rainforest right now, in the understory, 25 centimeters above the forest floor, an ant that was walking normally 2 weeks ago is making its final approach to a leaf it cannot identify as its destination. It does not know it is infected. It does not know it is being directed. It does not know that the fungus inside it has been managing its body since before it began behaving strangely, since before it started leaving the colony at unusual times, since before it began falling from surfaces and moving toward the ground. And somewhere in the colony above, in the canopy where healthy ants move through the afternoon light, foraging and communicating and performing the 10,000 tasks that keep the superorganism functioning, there are ants that passed beneath an infected ant's dying body without knowing what was above them. They are carrying something home. The cycle runs. It has always run. And it will run long after the last researcher to study it has stopped asking how.